Welcome to This Explainer. Every single day there are these invisible, absolutely high-stakes medical wars being fought in hospitals all over the world. It's a silent battlefield where the enemies are entirely microscopic and the margin for error is literally zero. Today, we're stepping into the shoes of the microbial detectives who fight on these front lines. We're going to decode the forensic laboratory work required to track down two notorious and vastly different bacterial culprits, Brucella and MRSA.
Let's kick things off with a little bit of lab humor. Bacteria, the only culture some people have. Okay, jokes aside. In a clinical microbiology lab, culturing these organisms is deadly serious business. And this perfectly sets the stage for a deep dive into the microscopic world, where growing and identifying these cultures is absolutely key to saving lives.
Think of this explainer as our investigator's dossier. Here is our agenda for today. First, the diagnostic front lines. Then, profiling the microbes, methods of attack, identifying the culprits, and finally, neutralizing the threat. Let's get into it.
Section 1, the diagnostic front lines, where the investigation begins. We're looking specifically at the microbiology unit of Al Sabah Hospital. The reception process here is incredibly rigorous. And why is that? Well, because if the lab mismatches the patient identifiers right at the start, the whole forensic investigation falls apart before it even begins. They have to verify at least two matching identifiers on both the request and the sample, like a civil ID and a name. From there, they generate a unique barcode, distribute the sample, and actually rematch and register it again in its specific section. It's an ironclad chain of custody.
Section 2, profiling the microbes. Introducing the culprits.
1.4 billion. Let that number sink in for a second. That is the staggering number of cattle infected worldwide by our first suspect, Brucella. Brucellosis is zoonotic, which means it jumps from animals to humans, and it leaves this massive global agricultural footprint, causing severe human illness and huge economic losses, especially across the Middle East, Asia, Africa, and South America. We're talking about a global threat on a massive scale. Now, on the flip side, we have MRSA, or methicillin-resistant Staphylococcus aureus. Discovered back in 1961, MRSA is completely notorious. It's highly resistant to all available beta-lactam antibiotics, which includes your standard broad-class penicillins. And this resistance isn't some random fluke. It's driven by a specific piece of DNA called the MICA gene. This gene essentially alters the bacteria so the standard drugs literally cannot bind to it. It's a massive hospital-acquired threat today. Looking at them side by side really highlights the contrast here. On the left, Brucella is a gram-negative, non-motile coccobacilli. Basically, it's a short, rod-shaped bacteria that can't move on its own. It's an animal-born, stealthy, intracellular invader. On the right, MRSA is gram-positive, forming these spherical, grape-like clusters. It's highly contagious, spreads human to human, and launches really aggressive surface attacks. So we've got two completely different biological criminals that need entirely different diagnostic approaches. Section 3, methods of attack, their modus operandi. Let's see how they actually breach our defenses. So Brucella relies heavily on zoonotic transmission. It does not spread from person to person. Instead, humans have to step into its cycle. Usually this happens if you consume raw, unpasteurized dairy from infected cattle or sheep. But it can also invade if you inhale contaminated air, which is a huge risk for slaughterhouse workers or lab techs, or just by touching the fluids of infected animals if you have abraded skin. Once it's inside, Brucella's method of attack is just incredibly sneaky. It acts as a cellular intruder. Instead of hiding from our immune system's frontline soldiers, the macrophages, it deliberately lets itself get swallowed up, or phagocytosed. But it doesn't die. It uses its unique biology to survive and replicate right inside the macrophage's internal compartments. It literally uses our own security guards as a hideout. Furthermore, it targets dendritic cells, essentially hitching a ride across the body's tissues. These infected cells carry the bacteria deep into the lymphatic system, the liver, and the spleen, where they set up shop, forming these destructive focal abscesses and inflammatory granulomas. MRSA, however, takes a much more direct opportunistic route. It thrives in crowded environments. Hospitals, nursing homes, those are its prime hunting grounds. It exploits direct skin-to-skin contact, sneaking in through minor cuts, scrapes, or by exploiting a weakened immune system. Once it establishes a foothold, it causes these severe, painful, pus-filled boils and abscesses,
And if it pushes deeper into the bones or lungs, we're looking at really deadly conditions like pneumonia.
Section 4. Identifying the culprits. Under the microscope. This is the core forensic work. How do our detectives actually see what they're fighting? Visually, when cultivated on blood agar plates, the lab sees stark differences. Brucella is notoriously slow. It can take days, sometimes up to 4 weeks in severe cases, to form these small, translucent colonies that don't even break down the blood in the agar. It requires a lot of patience. MRSA? It's fast and aggressive. It grows really quickly at human body temperature, churning out these distinct, striking, golden yellow colonies that physically break down the blood in the agar. What's truly fascinating is MRSA's sheer environmental resilience. To isolate it, the lab uses mannitol salt agar. Get this, MRSA is so salt-tolerant, it can survive in environments with up to 28% salt. That's exactly how it thrives on the salty surface of human sweat. When it ferments the mannitol in this medium, it produces an acid that turns the whole plate this brilliant, unmistakable bright yellow. It instantly exposes its presence. To be absolutely certain, they also run a coagulase test as a definitive biological fingerprint. This test detects an enzyme that causes blood plasma to clot. If you see visible clot formation in the tube, well, it successfully separates dangerous Staph aureus, like MRSA, from other harmless staph strains that might just be hanging out on your skin normally. To complete the physical identification, microbiologists use some pretty neat rapid screens. For Brucella, there's the Roux-Bengal test, a quick, affordable spot agglutination test, where clumping means a positive reaction to Brucella antibodies. For MRSA, a direct microscopic exam reveals its classic gram-positive structure. It looks exactly like deep purple spherical cells huddled closely together in tight, grape-like clusters. But here's the thing. Sometimes traditional cultures just aren't enough, or they take way too long, especially with a slow grower like Brucella. That's when modern labs pull out PCR, basically a molecular search warrant, to amplify the bacteria's unique genetic code. For Brucella, they look for the IS711 insertion sequence, which is totally unique to it. For MRSA, they hunt down that infamous MESA gene we talked about earlier, the one responsible for its severe resistance. Section 5. Neutralizing the threat. Treatment and prevention. Once identified, how do we stop them? Defeating Brucella really comes down to strict agriculture safety and prevention. You have got to avoid unpasteurized dairy and ensure ground meat is cooked to at least 71 degrees Celsius. Farmers and vets absolutely must wear protective gear, like rubber gloves. If an infection does happen, well, there's no human vaccine, so it requires a really extended course of specific antibiotics, like doxycycline and streptomycin, for 2 to 3 weeks, just to root out the bacteria hiding inside your cells. Combating MRSA, on the other hand, is all about rigorous hygiene, frequent hand washing, keeping skin wounds covered up with clean bandages, that sort of thing. In hospitals, it's about strictly enforcing infection control protocols to isolate outbreaks and protect vulnerable patients. And since MRSA totally laughs at standard penicillins, doctors have to use specialized, heavy-duty, targeted antibiotics, like vancomycin or linezolid, that can actually bypass that beta-lactam resistance. As bacterial resistance continues to evolve, will our laboratory detective work be enough to outsmart the next wave of invisible threats? That is the million-dollar question. We've seen the incredible, forensic lengths microbiology labs go to, using chemical tests and genetic profiling to hunt down enemies like Brucella and MRSA. But these microbes are ancient, adaptable, and constantly mutating. It's an ongoing, high-stakes arms race between human health and microscopic evolution. Thank you for joining me on this explainer, and remember to keep your curiosity and your hands clean.